Okay, class begin. This is the last times for the stereo class. I later my students the 2 days free and reviewed all of the chapter. We already finished that, included the structures, the metabolic and the enzymes, it is on. All of this you finished, and this chapter maybe concluded that all of the contents already we finished. And summarize the characteristics about the metabolisms, including the metabolisms of the carbohydrate, metabolisms of the lipid, and metabolisms of the protein, the metabolisms of the nucleic acid, and all kinds of the substances you can see interrelated. So we summarize all of this chapter in this chapter. Okay, and first you can see summarize the speciality of the metabolisms in the human beings. Here you can see the first, you can see the metabolic process of the various substances, lipid, carbohydrate, protein in the body are interconnected and two forms the whole. And the process, like you can see, the digestion, absorption, intermediary metabolism, and last forms the waste, carbon dioxide, and so on, may be expelled from our body. So all of this you can say forms the wholity. And the second, you can see the metabolisms of the substance in the body is constantly finding regulated. And this we start every pathway. You can see finding regulated. Or like this, you can see the internal external environment is changing, influence on the body's metabolism. So the body has fine regulatory mechanisms to regulate the intensity, direction, speed, and last make the individual, make the organisms, make us adapt to the change in the environment. So this is very nice to adapt to the environment. For example, and also you can see you feel the temperature increase, and that times maybe our body maybe regulate the energy released and keep the body's temperature. And another one you can see each tissue and the organ, the substance metabolisms have its own characteristic. This we studied, you can see, for example, gluconeogenesis, only finished in the liver, only finished the liver and the kidney. Why like this? Here you can see different tissue, different organ have different structure, have the different kind of the contents of the enzyme system. Now, for example, the gluconeogenesis. Why? Only finished in these two organs because have the coding enzymes. And for you can see the all metabolites have their common metabolic rules. For example, we studied the glucose. You can see here from the food, glucogenesis, gluconeogenesis. All of this you can see can be produced the glucose and forms glucose pools. For example, another one you can see the amino acid pools. You can see can be synthesized the protein hydrolysis. It is so on can be produced the amino acid. And finally, you can say ATP is the common forms of energy stored and consumed by the body. And you can say nutrient, oxidated decomposition, and oxidated thoroughly, and produced ATP. And the last, you can see always NADPH provided the reductive equivalent required for the metabolisms. For example, this I summarize. You can see this is an example. The pentose, most of the NADPH come from the PPP. You can see used to synthesize fatty acid, used to synthesize the cholesterol. This is the sixth property for the substance metabolisms in our body. Okay, and second section, you can see the metabolic interrelationship. You can see the metabolisms of the various energy substances are interrelated and interconnected. Here you can see three nutrients: the carbohydrate, fat, and the protein. You can see can be oxidized to produce the energy in the body, and with the common pathway through cycle and oxidative phosphorylation, and forms the common interproducts. From this point, And we can lose the energy supply. The three nutrients can be substituted for each other and restricted each other. In general, the body's priority for fuels use are glucogen and fat and protein. The main energy supplied to sugars and fat, and try save the protein consumption. And the second, you pay attention to the metabolic predominance of the any energy supplying substance often inhibited and saves the degradation for other substances. For example, the fat, the lipolysis increased, and ATP produced increase. At that times, restricted the carbohydrate metabolism. And inhibited the 6-phosphofructose kinase 1, the number one in the glucolysis. We studied already. So at that times, decomposition of the sugars is inhibited. And another example, you can see when hungry, the short times the short days, 1-2 days hungry. At that times, you can say glucogen control and the muscle glucogen breakdown increase. And at that times, proteolysis increase. Maybe long times, 3 to 4 weeks, you can say the main energy supply is the composition of lipids and the ketone bodies. The protein breakdown was significantly reduced. You can say 1-2 days, you can say proteolysis increase, glucogen's breakdown increase. And the second, you can see the carbohydrate, fat, protein metabolisms are close interlinked by the intermediate metabolites. You can see in the body, sugar can be converted to the fat. We studied in the unit about the metabolism of the carbohydrate. We lose, you can see the sugars can be converted to the fat. But here you remember the fatty acid can't use to produce the sugars. There may be something saying the glycerol can be used too, and normally mobilized and glycerol may be oxidated thoroughly, release the energy, all used to produce the protein it is all, and can't used to produce the sugars. When sugars intake exceeded the, here you can see, exceeded the energy expenditure, and sometimes you can see glucose can be used to produce the fat. Maybe another pathway you can see synthesize glucogen storage in the liver and the muscle. And the second, you can see glycerin part of the fat can be converted into the sugar under special condition. Glycerol can be used to produce the glucose. under special condition. The fatty acid through the beta oxidation and produce acetylcholine can't be used to produce the glucose. And the third, you can say, the metabolism of the fat is influenced by the glucose metabolism. For example, here, starvation, and sometimes glucose supplies is shortage, impede the glucose metabolism. And at times, you can say fat mobilization increase, and the ketone bodies produced increase. And at times, lack of the carbohydrate. At that times, you can say the oxidation, the ketone bodies oxidation is restrained, and that times make the hyperketonemia. And second, you can say the part of the carbohydrate in the body and the most amino acid compounds shape skeleton can be converted to each other. For example, here adenine can be used to produce the glucose, converted into the carbohydrate. And second, intermediate of the glucose metabolisms may be aminated, can be produced the amino acid. Here you can see pyruvate. Pyruvic acid, you can say transamination can be produced adenine. Oxalose acetate can be produced aspartate. And the alpha ketone glutaric acid can be produced glutamate. And the lipid can't convert into the amino acid, but the amino acid can be converted into the fat. You can see the first, you can see protein can be converted to the fat, and second, you can see we already studied. You can see the amino acid. You can see deamination can be produced different kind of the intermediate products. Here you can see, for example, the acetylcholine and easily used to produce the fat. And second, you can see the amino acid can be used as the raw materials for the synthesis of the phospholipids. You can see sterins can be used to the phosphatidylase list. And third, you can see the cholesterol part from the fat can be converted into the non-essential amino acid. So if someone says that fat can't to convert to the amino acid, is wrong. You can say the fatty acid can't to convert to the amino acid, but here cholesterol of the fat can be used to produce the amino acid. And last one you can say is however, it can't be said the lipid can be converted into the amino acid. This is the partially right. Okay, and both you can say the certain amino acid are precursor to synthesize the nucleic acid. This pyrimidine purine we already finished. You can see the purine synthesize. You can say glycine, aspartate, glutamines, one carbon unit. It is wrong. Pyrimidine you can see aspartate and the glutamine used to synthesize nucleic acid. Phosphate reverse is supplied by the PPP. Here you can see already we finished in the amino acid metabolisms. You can see different kind of the amino acid. You can see can be produced different kind of the intermediate products. Acetyls, coenzyme. Here first you can see the fat produces fatty acid, the glycerin, stammering, and can be produced the uh, intermediate products. And another thing is you can see acetylcholine can be used to produce the fatty acid, can be used to produce the glycerols, ketone bodies, and all kinds of the amino acid. You can see can be produced the different kind of the intermediate products. Pyruvate acid, for example, the aniline deamidation produce the pyruvate acid. All of this summarize this from this figure. And furthermore, all of the contains in third section in this chapter. You can see the several important organ, the tissue, summarizes the property and the relationship of the metabolisms. First, you can say liver. Liver is the most important metabolic center and the heart of the human being. It plays the unique and important roles in the metabolisms of the sugars, fat, proteins, waters, and in organic salt, vitamin, and so on. For example, the rules of the liver in the glucose metabolism synthesize the glucogen, its main organ of the gluconeogenesis. This all kinds of the pathway we already studied. So the liver play important role in the maintaining blood glucose stability. We all finished that. And second, the heart. Heart, you can see, can use a variety of the energy material, mainly the aerobic oxidation, the ketone bodies, lactic acids, free fatty acid, glucose, it all so on. The ADP is produced by use the liptic acid as a fuel. The energy can be supplied in turns by the consumed energy material, such as the free lipids, glucose, ketone bodies. Third, brain. Brain, you can say, main use the glucose for energy. It consumes much energy. If the energy supply is not enough, at that times you maybe feels low sensitive to all kinds of the reaction. Brain consumes a lot of the energy and the oxygen. And the glucose is the main energy source. Consumes about the 100 grams per day. Lipid acid can't be available, and ketone bodies can be used for the uh, energy supply in the short supply. And third, you can say muscle. All the times we studied, you can say the muscle mainly oxidizes the fatty acid. And the intensive exercise produces the large amount of the lactic acid. You can see synthesized and storage of the muscle glucogen. Oxidations of the liptic acid in the main energy supply. And the last, you can see here, glucolysis is the main pathway supplies energy for the battery red blood cell. Without the mitochondria, the battery red blood cell have no mitochondria and consumes 15 to 20 grams glucose every day. And last, you can see adipose tissue is the important tissue for the synthesis and the storage fat. Important tissue for the synthesis and the storage of the fat. To decompose the fat into the fatty acid and the cholesterol for use by the other tissues for the body. The kidney is another organ for both gluconeogenesis and the ketone production. Renal's medulla is main powered by the glucolysis. The renal cortex is the main supplied by the aerobic oxidation of the lipids or ketone bodies. Here, this PPT, you can see summarize livers, brains, heart, adipose tissues, skeletal muscles, kidney, red blood cells. You can see all kinds of the tissue organs. You can see the specific enzymes location, distribution, and the function. Major metabolic pathways, the energy supplies, the products. All of this summarized in this table. Mm. For fourth section, reach the 10 minutes. We continue. This part, you can see different label and focus the different enzymes and organ and regulated. Okay, reach the 10 minutes.
Okay, Lasha.
Okay, continue. Okay. This is the last section in this chapter. Maybe some my students already remember about the regulation. For the every kind of the metabolic pathway. For example, glucolysis, you got the ATP, how to regulate that. So this except the regulation about the activity of the enzyme and another things for the advanced organisms, the regulation in your physiology, you study something. The levels regulations, the body liquid regulation, body the regulation of the body liquid. This mainly it is complex. So here you can say the regulation for the single cells organisms regulation. You can say metabolic regulation is an important feature in biology. For the single cells, you can say regulation mainly regulated the enzyme's activity and the enzyme's concentration. Through the changes the metabolic body concentration in the cell, which is the cell's the primary regulation. For the organisms, for the advanced organisms, on the cell level, also abide by this kind of the regulation. Enzymes activity, the enzymes concentration. So here, advanced organisms three levels metabolic regulation. First, cell levels metabolism. Same to the single cell. And second, you can see the harmless level. Just I emphasize for you, you can see mainly it is the harmless. Endocrine cell and the endocrine organ have appears harmless secreted by enzymes can play important roles and regulated the metabolism. And last, you can see the overrules horizontal metabolic regulation under the control of the central nervous system. The target cells and directly affected by the liver's fibers and the neurotransmitters. The metabolism and the function of the certain cells are regulated by the secretion of the certain hormones. For example, insulin secreted into the blood can be regulated as the glucose level. At that times, why secreted the hormones? At that times, you can say the liver still and the neurotransmitter regulated the pancreas cells can be secreted the insulin. So at that times forms the overrules horizontal metabolic regulation. On the cell level regulation and regulated the activity of the case that this we already studied all of the metabolisms of the carbohydrate, the carbohydrate and the lipid and the amino acid and the nucleic acid, it is on every pathway. We already, you can say, regulated the activity of the K enzyme. The intracellular enzymes are isolated and distributed. The speed and the direction of the metabolism pathway determines the activity of the K enzyme. And that times you can see feedback. You can see by the many principles. I summarize those rules. You can see the upstream products always act as the activator, and the downstream products, it is the inhibitor. And like this, you can see the still enzyme system and the isolated different region at that times increase the activity. For example, some chemical reaction finished in the mitochondria. For example, oxidative phosphorylation. The protein lysis in the lysosome proteasome. It's strong. Here you can see different chemical reaction located distributed different cellular region. You can see still cellars, the glucolysis, it is strong. Urea synthesize. I summarize in this table. All of this we already finished that. This kind of the distribution, what kind of the significance you can see, can be increased the enzymatic reaction. So the various metabolic pathway don't interfere with each other, coordinated with each other. This kind of method is conducive to the specific regulation of the regulatory of the each pathway. Another one, the metabolic pathway are a several of the enzymatic reaction whose speed and direction are determined by the K enzyme. Here you can see presented the several definition. The reaction catalyzed by the K enzyme and the following characteristics. The speed is the slowest. The catalyzed by the speed limited enzyme. This first gives you definition. What is the speed limited enzyme? Its speed determines the total speed of the entire metabolic pathway. So give this enzyme speed limited enzyme. And another one, the catalytic one-way reaction, irreversible and non-equilibrium reaction, which the activity determines the direction and the entire metabolic pathway. This is the K enzyme. So the enzymes having this kind of the characteristics, we see this enzymes in this metabolic pathway, it is the K enzyme. For example, glucose kinase, the 6-phosphate fructose kinase 1. It's all. It is the K enzymes in the glucolysis. And another one you can say is the activity of these enzymes is controlled not only by the substrate, but also by the avoidant metabolism and the effector. Now, also the enzymes can be regulated. Regulation, we studied the allosteric regulation, the cognitive modification. It's on. Here key enzymes list here. Glucogen degradation. Last you can see the phosphorylase. The glucolysis, you can see three kind of the key enzyme. And secondly, you can see metabolic regulation is mainly achieved by the regulation activity of the key enzyme. And at that times, you can see a low state regulation, chemical modification, combinative modification. This and the slowly regulation regulated and changes the concentration of the enzyme. As this, we already studied. So quickly finish that. You can say allosteric regulation. Here you can see summarize all kinds of the allosteric activators, inhibitors. You can see can be regulated the every, for example, Krebs cycle. You can say AMP, it is the activator to the key enzyme citrate synthers. All of this you can see. So give you 3 days or 2 days. Read this part. You can see all of this listed here. And another one, so you can see the initiator. All products of the metabolic pathway affect the metabolic pathways through the allosteric regulation. There's two parts, the enzymes and the effector, inhibitor, activator. Here you can see the allosteric effector and aided the regulation stability of the enzyme. Make the enzyme's advanced structure changed. At the three-dimensional structure changed. At that times, the enzyme's activity changed. This, what kind of the usage? Physiological significance. You can see the enzymes in the feedback inhibition response pathway prevented the metabolic tears from being generated too much. You can see here the long chain fatty acid coenzyme A. You can see act as the inhibitor and decrease the acetylse coenzyme carboxylase. Another one you can see allosteric regulation in make the efficient use of the energy without the wasting. Labor forms the infiltrate. You can see here, glucose 6-phosphate can be storage. At that times, you can see activity of the glucogen synthesis and promote the sugar's storage. Another way you can see at that time decrease the glucogen phosphorylase, inhibit the oxidation of the sugar. Third, you can see here allosteric regulation combines different metabolic pathway. Here, for example, the citric act as the activator and promote the fatty acid synthesis and inhibitors are 6-phosphofructose kinase 1 and inhibitors the glucolysis and inhibitors the oxidation of the sugar. Another thing is the activity of the key enzyme can be regulated. This we already finished. For my students, only remember the phosphorylation and dephosphorylation. You can see here acetylation, deacetylation, methylation, demethylation. And here my students only, you can say, summarize. We studied in our book, you can say, phosphorylation, dephosphorylation. What kind of the effect? For example, you can say, during the Krebs cycle, during the aerobic oxidation of the carbohydrate glucose, you can say pyruvate dehydrogenase, you can say phosphorylation inhibited. At that time, this dehydrogenase, you can see the activity decrease. When dephosphorylation activity, hey, you can see here focus on the phosphorylation, dephosphorylation is okay. Hey, here you can see phosphorylation, dephosphorylation, focused on the threonines, sterines, tyrosines. This three kind of the amino acid residue. The kinase, catalyze, and phosphorylation. And phosphatase, catalyze, dephosphorylation. Here, what kind of the characteristics for the chemical modification? The combinative modification of the enzyme's protein is a reversible enzyme's reaction. Under the action of the different enzyme, the active state of the enzyme's protein can be changed to each other. Enzymes are regulated in vivo by the regulatory factor such as the hormones. Another thing, it has the amplification effect, prostate, higher efficiency than allosteric regulation. Phosphorylation and dephosphorylation is the most common methods. The same enzymes be regulated by the both allosteric regulation and the chemical modification. This we studied during the we studied the metabolism of every chapter. And another one, you can see the enzyme's activity can be regulated by the changes the concentration, slowly regulation. One, induce. For example, every times maybe you take one bowl of the food for the dogs, and sometimes the dog maybe look their food coming, maybe begin to secrete their digestive enzyme. It's all induced. You can say the contents of the enzyme protein can be regulated by the induced or repressing the expression of the enzyme's gene. The common mode of the induction and the repression. By the first, you can say by the substrate, and another one inhibiting of the enzyme synthesis by the products, and third, induced the enzyme synthesis by the hormones, and last, you can say drugs. Regulation of the steel enzymes contains can also be achieved by the changes the degradation rate of the enzyme. Degradation, we already studied two kinds of methods: lysostome degradation and protostome degradation. You can see. Second, you can say hormones regulated metabolic process by the act on spatial receptor. Hormones, for example, you studied in the cell biology and the physiology. You can see when the environment changed and the times relative tissue of the body secreted the hormones. Hormones, you can see transported to the in the body, and the hormones bonded to the receptor of the target cell, And last, the target cell produced the coding effect and makes the bodies adapt to the change of the environment. Here you can see for the harmonies you already studied in the physiology. You can see some harmonies can be only transported the signal and bonded with the acceptor. Some more harmonies molecule may be transported into the cell and bonded with the intestinal receptor. Here you can see this are copied from the cell biology. You can see bonded with the membrane receptor and produces the secondary messenger molecule. For example, CMP and activity the other enzymes. It is so on. This you can see the harmonies directly transported into the cell and into the nucleus, and it makes the coding genes translation or transcription. And last, you can see make the genes translation. So here you can see the organisms of the whole regulated the metabolism of the substance through the nervous system and the liver harmonies pathway. So for the advanced organisms, you can see, and at times you can see sugars, the fat, protein metabolisms vary in different hunger states. For example, the short times hungry increases the fat mobilization and decreases the sugars usage, glucogen consumption. At that time, blood sugars tend to go down, decreases the insulin secretion, increases the glucogen secretion. And at that time, cause a state of the metabolic change. The first, you can say, the lipid metabolism change. Fat mobilization, stressing, and the ketone body production increase. And the glucose metabolism, you can say, sugars displeasure to stressing. The localization of the glucose utilization. For the protein metabolism, you can say, most protein breakdown is stressing. And amino acid are allosterogenic sugar and compensated the sugar shortage. For the chronic hungry, compared with the short hungry, you can see change in the protein breakdown reduced. For the glucose metabolism, you can see the liver and the kidney displeasure sugars enhanced the glucogen displeasure of the main raw materials of the lactate and the pyruvate. Another thing is for the lipid mobilization furthermore increase and brain tissue used ketone body increase. And another one, you can see under the stressing condition, stressing can be increases the energy supply of the sugar, fat, and breakdown of protein breakdown, and limit the energy storage. What is the stressing? You can say stressing refers to the state of the stressing in which the human bodies is subject to the serial response to the unusual stimuli, such as the tremors, steaming pains, and froster speed bite, lack of the oxygen, poison infection, and steaming mood swings. At that time, you can see under the stress condition, the whole holistic body respond. Here you can see sympathetic excitation, and at the same time increases the secretion of the adrenals medulla and the corticosteroids, and the glucogen and the growth hormones are increased, and the insulin secretion is decreased. Post steroid of the metabolic. Um, Under such condition, the metabolic change, you can see the blood glucose elevate and fat mobilization furthermore enhanced, and proteolysis enhanced. 怎么？你稍等一下，我码一下。And another thing is the proteolysis increase. So during these times, you can see different organ, different tissue, what happened. You can see what kind of the metabolic pathway changed. For example, the protein breakdown may be decreased in the muscle under such condition. During stress, liver, for example, the liver, gluconeolysis increase, glucogen synthesis decrease, gluconeogenesis increase, fatty acid beta oxidation increase, ketone plasma increase, and the last product produced the condition glucose increase, ketone bodies increase. Okay. Another thing is obesity is a disorder of the eating behavior and the energy metabolism caused by the many factors. These are some people, some students maybe have the deeply impressed metabolic syndrome. You can see this syndrome characterized by the opposite higher blood pressure, glucose metabolism, and the dyslipidism. On such conditions, there are different types of the fat storage in obese people. For example, the second obesity, you can say constitutive 6 is fat. At that time, the fat that adolescent period see more basically be caused by the result of the adipose cells number increase. The cell maybe proliferation increase. Another thing is acquire rate obesity, and at times you can say adult obesity caused by the excessive nutrition, and many due to the increase the size of the fatty cell, but also increase in the number. And the secondary obesity, you can say certain nerve or endocrine disease caused. So this in your physiology, you can say the body mass index. For this, you can say the commonly used criteria for the diagnosis. If these people it is the obesity or not, BRA. When this Obese are those who weigh more than 20% of their secondary body's weight, have the body's mass index above 30. It is the obesity people. And the second, you can see the balance of the normal appetite, itch, and energy expenditure is completely regulated by the livers and the endocrine system. Here, insulin and the leptin. Recent the years, leptin, the scientific research is very hot. And last one, the obese people often shows insulin secretion. Abnormal function and disorder of the glucose and the lipid metabolism. And the last topic, metabolic metabolism is metabolic mix is the studies of the smaller molecular metabolities and the global level. This is the comprehensive scientific research methods. Detect the all those molecular mass metabolites in the organisms or in the cell. The whole quality and doing scientific research. And second, you can say the study of the metabolic mix requires a higher strong pot quantitative detector technique and the large skills calculation. Here you can see list the nuclear magnetic resolution technology have the great advantage. This nuclear magnetic resonance is the most commonly used. And maybe in the clinical hospital also use this. And another one, simple component analysis is the most common used and effective methods in the pattern recognition. Another thing, metabolic component fingerprint can be obtained. For this, you can see the metabolic mix have the greater potential in new drug discovery and disease diagnosis to study the mechanisms of the action of the drugs and the function. It is widely used as a toxicity evaluation of the candidate drugs. And last, identified the vulnerable patterns of the disease-related metabolic biomarkers for the diagnosis of the disease. So this kind of the methods used the discovery of the new drugs and the disease diagnosis. So okay, and now this chapter we finished all of the contents we studied and finished. And my students pay attention to. For all of you, the theory class today is finished. This today you have this part maybe focused on the relationship and the comprehension of the metabolisms in our body. And left the experimental class. I think maybe waiting the next semester, maybe when you come back to China, maybe in the number rooms finish the experimental class. Okay, for this subject, left maybe 2 weeks and combined the ebook and the PPT. Reviewed this part again. Thirdly, you must finish all of the homework, the examination, the quiz, and maybe the medical school arranges the examinations times. Okay, today we stopped here. All of the class is finished.
OK 老师。
Okay, and for the experimental class, waiting for the next semester, we continue. The examination focused on the theory class. Okay, totally 30 chapter. Okay, today class is over.
OK 老师。谢谢老师。
谢谢姐姐。